Welcome back, everyone. And I'm Kwabena. I'm a PhD student at the Research Center ULISH Enabling Technology Group. And today I'll give tidbits of my PhD topic. And for the sake of today, I've titled my talk Syndrome Bus Research Disease Induces Heterogenic Allocation of Carbon in Growing Sugar Beet. So I believe the French word here, Syndrome Bus Research, which I'll be referring to as SBR, is quite new to most of my audience today. And before I dig deeper, I will introduce us to this French word and what it's doing to sugar beet plants. So SBR is a fast-spreading sugar beet disease basically in mid-Europe. And what the disease does is that it reduces absolute sugar content and biomass of affected plants by 25 and 5% respectively. The symptoms of this disease is quite remarkable for both above ground and below ground crop organs. And in above ground, we've observed some proliferation of new leaves and also yellowing of new leaves. But these symptoms become apparent at very later stages of infection. If we dig into the below ground section, we see some brownish discoloration in the vascular bundle. So if we slice into sugar beet of SBR infected plant, we see that portions of the cross-section show some brownish discoloration within the vascular bundles. And this is basically the main symptom expressed for sugar beet infected plants. And this disease is caused by two main so-called phloem-restricted plant pathogenic bacteria. And the first one is the Proteobacteria candidatus acinophonos phytopathogenicus and the Phytoplasma candidatus phytoplasma solani. So this disease is a so-called vector-transmitted disease. That means that it requires a carrier to transmit the disease into, to transmit the pathogens into host plants. So what this plant hopper here called Pentacillidios leporinus does is that it visits innocent plants, feed on them, and try to introduce the pathogens simultaneously into the phloem sap. So among several intricacies revolving around the subject matter of SBR, the mechanistic understanding regarding crops infected with sugar beet is currently incomplete. And since it's a phloem-restricted plant pathogenic bacteria, the trajectory of the carbon resources within the phloem and the development of the whole beet becomes an interesting niche to study. So to do this, we asked ourselves two questions. The first one, we wanted to understand how this disease alters structural development of sugar beet plants. Again, we wanted to have insight into how SBR disease affects carbon distribution dynamics in basically below ground part of the sugar beets. Now, for sugar beets, the below ground forms the main economic value, so it becomes important for this subject. So to do this, we set up an experiment in such microcosms. We grow sugar beet plants, basically one single genotype with two groups, the inoculated and non-inoculated group, to establish the disease infection. We confine young plants and the plant hoppers, which I've shown on the previous slide, in this kind of tent for an inoculation access period of 11 days. And through this 11 days, we hope that indeed the plant hoppers were hungry, furious, and fed on the plants to introduce the SBR pathogens in them. So before I go inside my results, I would like to give us an overview of the main phenotypic approaches I've employed and how I've achieved and analyzed my phenotypic data. So as I indicated earlier, we are interested in the structural and carbon allocation dynamics of infected plants. And to acquire structural information, we've employed a plant-dedicated MRI platform to non-invasively image basically the below-ground structural development of sugar beet. And for carbon allocation dynamics, we've employed a tracer transport technology and positron emission tomography to monitor or track the fate of carbon within the plant. So the business of the positron emission tomography is quite tricky. What we do here is that we supply a radioactive carbon C11 in the form of 11CO2. And the assumption here is that the plant will take up the 11CO2 and incorporate it into the photoassimilate. And then with the help of our PhenoPet platform, we are able to track the movement or the distribution pattern of the labeled photoassimilate within below ground of sugar beet. So after we acquire such expensive phenotypic data, what we do is to reconstruct those images into several projections, basically either two-dimensional projections or three-dimensional projections. And the next thing is to extract the phenotypic trait we are interested in. So from a single-handed MRI, we are able to extract structural information, basically the taproot, volume, and also quantify cross-sectional anatomical features. I will dig a bit deeper later. And again, another possibility is to co-register the MRI and PET images to have a more or less like a structural and functional perspective of how the tracer is distributed within the sugar beet. From this, the last step is to analyze the quantitative data we achieved from the previous step. So I will show some results on what we've achieved so far. So here I'm showing temporal development of sugar beet. Basically, these are the 2D slices of the sugar beet. So we kind of monitored the development of the cross-sectional features at 3 different spatial levels. So this is what we define as the upper section of the beet, the lower section, and the mid section and the lower section. We wanted to see how the disease affects the plant across different spatial sections of the plant. So here we've observed that in the healthy samples, we saw a clear defined anatomical features, unlike in the disease samples. You might probably appreciate it more if we have a closer look into the upper 2 slices at the last imaging date. So in a control sample, we see a clear defined anatomical structure for a healthy sugar beet. If any sugar beet enthusiast is here, he or she will disagree with me. This is how clear a healthy sugar beet looks like. But on the other hand, in the infected sample, the disease sample, we've seen that there was some distortions in the cambium ring formation, and there's a smearing of the annular rings. And also, if you compare these MRI intensities, we see that sections of the disease showed a brighter intensity than in the normal, healthy, cross-sectional sugar beet. So more on the structural data, we have quantified taproot volume as a direct measure of storage root development. And here what we did was to generate a binary image from the raw data we acquired from the MRI, and then again use a median filter to remove all fine structures like lateral roots, and the remaining orange I'm showing here define the taproot for us. So we first generate a binary image from threshold segmentation, and then we remove all fine root structures by applying a certain median filter and then generate a taproot from it. So here we observed that round about 49 days after inoculation, there was a reduction in beet volume, and this progressed until the last imaging days. Again, the next thing we did was to quantify the cross-sectional anatomical features here. So one unique feature about sugar beet is that in between these rings, cambium rings that I'm trying to depict here, is where the plant stores most of its sugar. So we try to quantify what happens there in terms of infection. So here we contoured just the 4 inner rings and applied a certain radial analysis to determine the width of the inner ring sections by ignoring or removing the central core. And here we also observed that as early as 42 days after inoculation, the ring sizes, basically the ring width, was reduced by the presence of the SBR pathogen. So The next session of my results, I will dedicate it to the tracer, what we've achieved so far from the tracer distribution dynamics. This was what I think Hendrik was trying to show in his talk, which he skipped, so I'm happy to fill in for you. So here I'm showing a video depicting tracer, the movement of tracer within temporal spatial slices of the sugar beet. So if you agree with me, for the control sample, we observed that the tracer filled most of the cross-sectional part of the sugar beet, but in the infect disease sample, we saw that some parts of the beet was receiving little to no photoassimilate, and these parts were also the part that was showing the bright signals as I've shown previously on the earlier slides. So here we observe a kind of sectorial distribution of the recently phased photoassimilate, in the sense that the part of the beet was receiving little to zero tracer. In fact, we didn't do this just for a snapshot. We traced back and we did this temporarily also. And here we kind of had indication of the sectoriality was already showing up at 42 days after inoculation, but it was really indeed very prominent at the later stages of infection. One thing I forgot to mention earlier on is that in these figures I'm showing, the gray part shows the MRI and the colored section shows the tracer we've applied. So the next thing we did was to estimate heterogeneity in the tracer distribution as an indicator for deviation from normal distribution. So normal distribution as in how homogeneous the tracer was distributed in the healthy sample in relation to what we've seen in the disease sample. So we did this for each sample, beet sample, and every circle I'm showing here represent 10 spatial slices. Like in the video, I was showing a couple of slices, but here we did it for 10 different slices for every sample. And we fit a linear model, and we observed that in the control samples, which I'm showing in blue, dotted lines, we didn't see any strong relationship, but in the disease samples, we found a strong relationship where we observed around 3% increase in heterogeneity per day in our measurements. So let's have a flashback here. Earlier on, I indicated we want to understand how SBR alter structural development of the beet. And indeed, we have seen that the disease is reducing the beet volume and also the inner ring width. And what I haven't showed today is the biomass, but we have a clear indication that the biomass is also reduced. Moreover, we've also observed deformation in the cross-sectional anatomical roots, anatomical features. Again, I wanted to know how the SBR affects carbon distribution dynamics in below ground part of the beet. And here we see that, as I showed in the video, there's a form of sectorial distribution of carbon in the presence of the pathogen. Moreover, we've seen an increase in heterogeneity in the presence of the pathogen. So from our results, we have a detailed catalog of how this disease is affecting sugar beet plants. And what we could do next from this is to use this as a basis for judging susceptibility and also selecting resistant genotypes to benefit us for future breeding programs. Again, it will be beneficial for us to also determine the shoot physiological responses during SBR sugar beet interaction, and this will provide us a holistic overview of structure and functional relationship existing between above ground and below ground parts of the beet. And one thing I would also want to emphasize is that the SBR disease is also extending its host range in mid-Europe. That means it's affecting other plants, and it will be beneficial for us to apply our systems into other pathosystems like in potatoes. I'm very thankful to the people I've listed here from the Forschungszentrum Jülich, Germany, and also the Institute for Sugar Beet Research in Germany. And thank you for your attention.
Thank you. We have time for a few questions. Well, okay.
Thank you for a very nice presentation. So I'm super happy to see the progress. Yeah. So just a quick question for you would be, when you do MRI scanning, right? You have a different time point scanning. So have you noticed any sort of effect on the pathogen side?
Of course, we are measuring sugar beet switches. So with MRI, we are basically measuring protons. That's the water in the samples, and the water content in sugar beet keeps increasing with time. So what we did was to tune and match our system to suit every stage of measurement. So of course, we have some little minute signal differences, but it doesn't really manifest in a negative way. Yeah.
Thank you.
I was wondering, have you also measured the starch concentration in the sugar beets, either by MRI or directly in a chemical way?
Of course, we could do this by, for example, chemical shift imaging, but we haven't established protocols for this particular niche of study. Yeah, but so it's a no. I haven't done that.
I expect the starch concentration has decreased, right?
I cannot conclude on this because if I think about the density of the infected and the control plants, I'm not assuming a lot of differences, but I will not say no or yes for now because I don't have a data. But if you ask me, I will say I don't assume a lot of differences in terms of it's not starch but sugar content at this stage, because we screen at the very infant stages of the infection. Maybe at later stages we will see more, but at this we've done our analysis until just 63 days after inoculation, and at this stage I'm not assuming more.
Yeah, over here. This carbon 11 stuff that you are working with, can you describe a bit how complicated it is to work with this material?
It's pretty complicated. So in Germany, we call something challenge shots. You need some radiation protection stuff to even, it's a full controlled area. So what you don't want to do, we produce, I think for each measurement, we produce 50 megabecquerel of the radioactive CO2, but we cannot be that exact. So we first trap it in a sieve and then put it in our system, and because it has a very fast half-life, it has a half-life of 20.4 minutes. We are able to do some algorithms to just admit just 50 megabecquerel in our system. But during this time, of course, we are not allowed to go there. We just keep the plants. All our setup, I think I showed the literature. If you are really interested in this, you can have a look in our setup. It's in a climate controlled area, so everything is under control. It's quite dangerous, but not that dangerous.
Do you think in UNIF we will continue working with carbon 11 in the upcoming 5 to 10 years?
First of all, I'm not a policymaker, but as a beneficiary from this, I will say yes, because we are not just using it for plants. There are I think Neuroscience Institute also in Ulysses has advanced system, and they are really deep into application of C11 in their systems. Yeah.
Okay. Yeah. Thanks.
Any other question? Yeah, let's thank you for running that.
And next up, we've got a last-minute fill-in. Thanks, Richard Poiré, talking to us about the Australian Plant Phenotyping Network.
Hi, thanks everyone. My name is Richard Poiré. I am the manager of the ANU node of the Australian Plant Phenomics Network in Canberra. And yeah, again, it's a last minute feeling, so I'm happy to present some of what we do in Australia. We are the Australian Plant Phenomics Network, and we set ourselves to be the partnering with various organizations for your plant phenotyping needs. We recognize that it's quite a complex subject, and that's why we in Australia also have a very fragile ecosystem. And it's been identified as a strategic direction 15 years ago that we need a network of coordinated facilities to tackle this challenge and provide our scientists and breeders with the right tools and the right expertise to do that work. So we used to be known as the Australian Plant Phenomics Facility, but as of this year, we are changed to the Australian Plant Phenomics Network because we went from 2 to 9 facilities, which warrants the change in structure and name. You can see on this map where the facilities are located. So we cover pretty much the entirety of the territory. And we have facilities that specialize in control environment phenotyping, 7 out of 9 of the nodes. We have facilities that specialize into the field phenomics. And we have central facilities also that help us collaborating together. We are collaborative by design, and it's not just a good intention. We also have people that are dedicated to make sure that we do collaborate within the nodes, but also outside. And for instance, data management is something that is tackled at the central level. So we have a team of people that make sure that the data we collect in every node can neatly interface with each other to enable meta-analysis and other work so that we make sure that the data is reusable. It takes a village to do all of this work, and it's not just my work I'm presenting here. So this is our team, for instance, at ANU. As of last year, we have a new director, Danny Way. Unfortunately, she could not make it to this meeting, but she came from Canada to start a position as a researcher, but also works as a director for the node. I am the manager, but then also within the team, we have expertise within project management, horticulture, biology, computer science, mechatronics, and biostatistics. It takes very much all of the skills to make it all work. And plant phenomics is quite complicated. I think the talks this morning showcase that. And we don't expect anyone to have all of the answers, but we provide a seamless experience by working together on that. So I will present some of the core offering that we are presenting, This is the very first plant I phenotyped a bit over 20 years ago. So it was slightly destructive. The plant didn't recover that, but I was harvesting plants, taking pictures of them, and then by hand measuring leaf length, width, and it's time consuming, gives you good data, but at the end of the day, it was not fast enough for the type of work we were planning to do. So we had to evolve towards more advanced technologies. And I think that's what plant phenomics has done over the last 20 years. And to be able to have a good phenotype, I think it all starts with a good control, a good environment that we can either control or monitor. So in our case, it's a collection of growth chambers that we have, like in different configuration that allow for different level of climate control, from the control, like the growth chambers, to the glass house, to more recently, we invested into growth capsules that are retrofitted shipping containers that have the latest technology in environmental control, whether it's temperature, humidity, multispectral LEDs. And some of them even have 3D multispectral scanners. And we also have tools that allow us to probe into more the physiology of the plant. We have thermal camera, chlorophyll fluorescence systems, and hyperspectral cameras and scanners that allow to look into more complex traits. So some of the chambers that we have, they cover ranges there from -10 to +40 degrees. So Australia has pretty extreme climate, and if you want to reproduce some of those climate conditions indoor, we need to have pretty strong control over that. Our facility is both PC2 compliant. That means that we can make and work with GM organisms, but we also have a facility that is quarantine compliant, which is very important. Australia is very protective with how it deals with quarantine laws. And if you want to import plants and work on quarantine material, it requires a certain level of containment that we do provide. We also specialize in dynamic climate control. So we do have rather intricate way to control the chambers. We don't just program like a day and a night temperature. We have ways to model the environmental conditions and plug that into the chamber so that you have the full dynamic change in temperature, light, and humidity at the 10-minute interval based on GPS coordinate of a condition, like a location we'd like to replicate, or artificial scenario when we want to induce environmental stress. We also have glasshouse space that people are able to access. We can control CO2, we can elevate it, and essentially make the plant grow into conditions that they are likely to experience in the next few decades. And we also have attached lab space that we can do PC2 work next to them. And more recently, we have 8 of those outdoor growth chambers, some of them with very high light capability, some of them with multispectral capability, and 2 of them with multispectral scanners that we can use to monitor the canopy growth over time. But it's not just the environment. We also have like quite a bunch of portable equipment that people can take from us and do some measurements with. One of the workhorse of our technologies, for instance, at the moment has been the use of portable hyperspec equipment. People use that in the field, in the lab. And 2 years ago, I was presenting a model that we've done on how to use this system to predict photosynthetic capacity at a much quicker rate than what we were able to do previously. We also have access to various hyperspectral scanner instruments. We have 2 of them at the moment. Chlorophyll fluorescence systems that we keep up to date regularly, but also coming from the physiology and ecophysiology side, we also have access to a range of gas exchange systems to measure plant photosynthesis. More recently, we've procured parameters that people can deploy in the field, and they're very easy to use, and they do both the colorimetry and the fluorescence in pretty much one click. And thermal cameras that we've been using quite extensively. And again, it's not just the kit, it's really the expertise. And when people come and reach out to us, they often have a scientific question but not know which tool to use, which one would be the most suited for their scientific question. So I think it's been a journey with them to make sure that they have access to the right tool, the right expertise, and design the experiment properly. And yeah, there's a lot that we also do with in the recent years with the 3D phenotyping of the plants. Most of the plants that we phenotype these days are hopelessly 3D, and just a top-down picture of the plant is not good enough to resolve the architecture of the plant. So that's why we've been investing in 3D canopy scanners, and this is the first Phenospec system that we had here. We have a second one now with the more modern sensors. They work very well, and they've been quite absolute workhorse of our recent experiments. They allow us to measure plants up to 1.2 meter in height, and the container that they are growing in goes all the way to 2.1 meter. And they generate both the point cloud and the CSV file that people need for their publication needs. So this is one example of the point cloud that can be generated on wheat plants. It's highly resolved, like we get sub-millimeter accuracy on on those, and we can collect traits based on two fields. Both we have a range of structural traits that revolve around biomass, plant height, leaf area, and a 3D leaf area. And other parameters, but we also have spectral traits that are more linked to the color of the plants and how we assess their greenness, the way we can bin the pixels into the pale green, the light green, the yellow, the brown, and also a range of indices based from the remote sensing community. But we also do work in the field, and I think I can't understate like just how much work it takes to coordinate a team of people working across Australia. So we provide field logistics. We've been training teams of people doing physiological measurement in the field. More recently, there's been a GRDC heat tolerance campaign that we've been coordinating. So it really involves training all of the people involved in the acquisition of data in the field to make sure that they collect the data the same way, they have good experience on the instrument that they're working with, and coordinating the field and working in the field with various weather conditions. Field campaigns that have to be postponed occasionally is quite hard. But yeah, our team has been doing very well there. And a couple of case studies of work that we've done in the past. We have worked with a ag-tech company who makes plant-based vaccines, so growing vaccines into plants using recombinant transformation of the plant. And they approached us to see whether we could find ways to better determine the yield predictors, so how to assess how much protein there is in the plant. And that protein is completely invisible, and unless you assess the plant by destructive measurement, you have no idea of how much protein of the compound of interest you have in the plant. So we've designed this high-resolution platform to reconstruct quite faithfully the architecture of the plant that they're working with at a leaf level. So we have leaf segmentation, that algorithm that we've developed to can identify the plants, and we've been able to track essentially and the history of the plant through the phenotype, and being able to identify which ones would be the most suited for the subsequent transformation, with the intent later on to use this 3D mesh as a way to map the hyperspectral traits that we've been developing. And another project that we have going on at the moment is as part of Australia's Economic Accelerator. Molecular farmers really need tools to be able to better assess their protein yield in plant. And one of the projects that we've been developing this year is testing a range of construct and transformation in the plant and phenotyping them with a hyperspectral scan to identify and quantify how much protein we have in the plant at different time points. We're still training the model at this point. We are still having to do the destructive measurement, but by the end of the year, we'll have a trained model that we'll be able to predict just by the hyperspectral signature how much protein of interest we have in the plant when it grows. One of the emerging directions that we are taking at ANU is also in looking into the hidden path and the root development of the plants. So we've procured 100 rhizo boxes. So they are pretty tall. They're like 1.2 meter tall rhizo boxes. So we have them already, but now at the moment we are designing the frame and the cassette that will be enabling us to do the phenotyping of those. So it's being fabricated at ANU in our workshop at the moment by the team of engineers. And within the next few months, we'll have a working system where we can water the plants and take out one by one. It's a semi-automatic system. It's not a fully automated one, but allows us to work with our rhizo boxes. We are looking into structural traits, but we're also looking at doing hyperspectral imagery of the plant and being able to collect exudates and have a bit of a better idea of how the physiology of the roots and how they're developing underground. But we also do develop a lot of custom software solutions that we are more than willing to share with people if they're interested. We've noticed that a lot of our users were doing a lot of that manually themselves, and we thought that there has to be a better way to do it. So our software engineer developed a first tool called Leaf Analyzer that replaces the need to use flatbed scanners or a conveyor belt system for the measurement of detached leaves. So it's based on a target that has QR codes attached to it. So all it takes is a smartphone or whichever camera of choice you have to take a picture of those. And the software does automatically the segmentation of the background, the foreground. It works with very difficult lighting conditions and report automatically about like the type of metrics that plant biologists are usually keen to get. So this is an example of what it looks like. So the left side is the raw pictures, and the right one is geometrically corrected and has the background removed, and it automatically saves the metrics, the color, the size on a CSV file that people can use. So yeah, that software is ready to go, and if people are interested in using it, we're more than happy to share it. It works with leaf, but it works with pretty much any material you can think of. It like any plant organ that is not white will be segmented pretty decently with it. Works with also to count objects, and it really takes no input from the user, so it's very fast and very user friendly. Another recent development that we've done is using regarding the segmentation of objects, and especially when you're looking at hyperspectral imagery, there's not much in terms of tools available to work in hyperspec dimensions. So we and Tao in our team created this software that allows us to segment pretty much anything with no click if it's very easy, or with a few pointers if the image is quite complex. So let me load a demo of how the system works. So by loading an image that was previously recorded, and all we need to do is identify essentially a region of interest that so some cropping within the image. The system will do a first guess of what the object of interest might be, and in that case, it's doing a good job at identifying what we want. But it's not. Sometimes it's not perfect the first go. So what Tao is doing here is placing a few pointers about like, yes, this is what I'm interested in, are the green dots, and the red dots are the one that, no, this is not exactly what we wanted. So the system will automatically correct the prediction and account for the user input when they are required. It depends on the complexity of the image really. And working with complex root images, sometimes it's very useful to have those, and automatically then it will save all of the metrics of the objects that have been collected there. So we intend to use this software as a first pass to segment the foreground, background in our riser boxes. So this is an example here, and it works very well even with very complex soil and architecture. We're in the process of improving that even further. And the last piece of software is application is with regard to hyperspectral imagery. As I mentioned, there's very few software that allows to do that. So what we have here is a plant that has been scanned through our hyperspectral scanner, but usually it's quite difficult to work with those images. So what we have here is a single click essentially to identify where the plant object is. The system will make a guess of what the plant is, and it will propagate the mask onto all of the hyperspectral layers and save the image as essentially a binary masked hyperspectral file that will allow for very much easier subsequent work and predictions on all of the pixels. So essentially each pixel becomes its own full hyperspectral scan, and we have a range of hyperspectral prediction model based on those data. So and that cleans up the data and speeds up the work a lot. So I think that's pretty much all I had this morning to present. But yeah, if, by essence, we are highly collaborative. So everything you can see here works for us, but our facility is really looking forward to collaboration with academics, industry, both within Australia and internationally. So if you have any interest, I think there's quite a few of us here. There are 6 of us from APPN. So yeah, reach out to us, and I'm sure we can find ways to work together. Thank you.
Thanks, Chuck. Do you have any questions? Good.
Hi, thanks for presenting the work you do on the other side of the planet. And a question regarding the segmentation tool you showed. is there also an option to have, for example, if you have loads of images, like hundreds of thousands of images, that you can pre-select and kind of train it or do a profile that you then apply as in an automated pipeline as batch processing?
Yeah, it does batch processing. Yes.
I just thought, is it based on one of the segmentation tool?
Is the segmentation tool based on something like one of those meta tools or something like that that's available, or did you all make something?
No, I mean, those are using pre-trained models, but yeah, we package that into something that is very, very easily usable.
Okay. And then was it trained specifically to work better on in this use case, this sort of use case, than if you just took the plain?
No, it works pretty much with any image. Actually, a lot of our team members use that to do snapshots of things on the internet and grab logos and things to copy paste. Like it really works on anything. It's agnostic. It's not just plants. Works on anything.
Yes, thanks for the talk. And I was indeed also interested by the software which you developed. How does this relate to the backbone of, for instance, something like Plant CV? Can you share this through a Jupyter notebook or a Plant CV backbone such that we all can try and take advantage from it?
No, we would be very happy to open source it and make it part of Plant CV. Like especially, I think the Hyperspec module is quite novel, so I think there's value in having that. It came from like the specific needs that our user had, so we developed that. But we realized that there's more wider appeal for this kind of software. So yeah, happy to discuss how we can do that.
Yep, that could be. So we are also working on some segmentation tools, some labeling tools, some AI training environment. It's not a question, actually more a suggestion. We got this new IPPN AI working group. I think we should align on what we are doing, because it feels a bit that we are doing similar things in parallel, and we don't need to have the best tool. We should have a common tool and speed up the developments.
I do agree, and I think for me, one of the highlights of coming to this meeting is seeing just how many networks of infrastructures we have, but like sometimes it's very hard to work collaboratively. We all want to be collaborative, but I think within APPF we are fortunate enough to have like a central team that makes sure that we are collaborative. But when we are talking about networks of networks, like what's the vehicle to make sure that operating procedures are saved somewhere, accessible for people? Where do we find software that people can use and reuse? More than happy to explore what that could look like.
Thank you for your presentation. My question is this software only working for leaves threads, or we can segmentation fruit, flower, and nodes, and another part of the plant?
Yeah, any organ. It's not optimized for leaf. No, it's it works for leaf, and it will generate parameters that are meaningful for like length and width that usually make sense for leaf tissue, but would work equally as well fruit, as long as they're not white.
Okay, thank you so much.
Gotcha.
Okay, now next we have the flash talks for the poster presentations. So before we start those, if all of the presenters can come and stand or sit somewhere near the front, then we can quickly go through. Okay, well the first one is mine, so I'm in the right place. Is everyone else who's presenting ready to come up afterwards?
Yeah.
So, I'm a postdoc at ANU. I work with Justin Borovitz, and I wanted to quickly tell you about a project that I started this year. The aim of the project, well, it's really motivated by global forest dieback. So across the world, as in Southeastern Australia, many forest ecosystems are experiencing demographic decline from combinations of abiotic and biotic stress. Often it's complicated and poorly understood. One of the hopes is that if we can phenotype individuals across forests, looking at canopy traits and canopy conditions, to get a look at the variation in stress responses, we could then go down onto the ground, sequence the genomes of selected individuals, and try to tease apart the environmental and genetic determinants of stress response. So we haven't got there yet with this project, but we're working towards that, and we're at a stage where we can start looking at individual tree time series, and I'll show you a pipeline that we've developed towards doing that. So here we've got LiDAR flight data from across the entire ACT, which is a few thousand kilometers squared. So this is 18 points per meter. And with that, we've been able to segment the tree crowns of 5 million large trees. An example of that is shown in that top middle plot. So for each of those, well, we're fortunate that 100,000 of those tree crowns we can assign species labels to through some field surveys that were conducted. So we have this. So next, we actually extract PlanetScope CubeSat data, which is high-resolution imagery almost every day with 8 bands. So we've got this time series for every single tree, multi-band. And that's really nice to start exploring species classification. For an example, I've shown some summaries of time series for oak species that are planted in around Canberra and 3 eucalyptus species. So you can see the variation between these groups. Obviously, oaks lose their leaves, but we're really more interested in the variation within species. So to what extent is this caused by genetic variation, environmental variation, and just noise in the data? And so that's kind of what I'm interested in talking about today. If anyone is interested in phenotyping trees using space, come and find me. My poster was actually yesterday, but I'd be really happy to talk to you all about it. Thanks.
Good morning. I'm Etsushi Kumagai from Nara, Japan. It's an honor to be present our work today. Biomass and leaf area index are key factor for crop yield. Typically measured by destructive sampling method, like this. Like this picture. So feature time consuming and labor intensive. To address this, we use hyperspectral reflectance as a non-destructive and faster alternative. Widely used full spectral band model like partial least square regression as a gold standard, but can lead data redundancy. So we try to apply the band selection technique to streamline process and improve the prediction accuracy. We collected the time series data of canopy scale, reflectance, biomass, and leaf area from many soybean genotypes across many years. Addition to PLS, and we also evaluate two band selection technique, bootstrap lasso and ensemble running. These methods allowed us to select the most important one for accuracy. This figure shows the PLS using all spectral band shows the highest accuracy for area predicting. However, ensemble running and bootstrap lasso using a pure band performed well. we conclude that both bootstrap raster and ensemble learning present efficient alternative of PLS by simplifying data complexity and maintain prediction accuracy for LAI. This selected band for prediction LAI could contribute to advances with multispectral sensor for agriculture monitoring, I believe. Thank you for your attention.
Hello, everyone. So microRNA seem to be a very new term and important term right now because of the news. The Nobel Prize for 2024 is given for the discovery of microRNAs, and microRNA play a very crucial role in my analysis. And so here, basically what we did was, so the questions that come forward in my analysis is, what are those genes that regulate the flowering time in sorghum? And flowering time being a very important trait, because it affects the yield and a lot of other things. So we are here trying to understand what are those genes that are affecting flowering time, what is the pathway that regulates flowering time, and how different is sorghum when compared to other grasses like maize. So here we are taking a very big population of 900 genotypes, and this population we collected the phenotypic data and scored flowering time for it. And we collected tissue data that was collected only in like 2 hours for this complete field by my lab mates. So first picture shows the diversity of sorghum that we had, and second picture shows that I was collecting the tissue. And yeah, so 2 hours was a very small time because RNA seems to change with time. And next is these tissues that were collected were then, we created a gene expression for it, identifies analysis, and a lot of genes that we can see here, the FT, the MADBOX genes, and these genes are actually regulating the flowering time in sorghum. So the next thing is, what is that pathway that regulates flowering time? So here it is the microRNA that is actually playing a very crucial role in the aging pathway of sorghum. And if you want to know more about this, please come and visit my poster, which is 52. I would love to talk about it. Thank you so much.
Hello, everybody. My name is Mildred Mayorga. I am a researcher at the Alliance Biodiversity and International Center for Tropical Agriculture. Okay. Pasture holds significant potential for sequestering carbon in soil due to their extensive coverage and their ability to allocate a substantial part of photoassimilate below ground through their root systems. Deep roots are an interest and particular interest because they can transport organic carbon to soil layers where it remains more stable. So, with the objective to identify tropical forest grass genotypes with root traits that promote soil organic carbon accumulation, an experimental trial was conducted involving root phenotyping of 180 genotypes grown in cylinders filled with soil. The evaluated genotypes included accession and hybrids of 5 different species of tropical forest grasses. Root phenotyping was performed using root images that were segmented and processed using a root image analysis software to quantify morphological traits. Additionally, we evaluated biomass distribution and an indicator of root depth. We identify 22 top-performing genotypes clustered by root traits, demonstrating increased root length, area, volume, high root-to-shoot ratio, and deeper root systems. Thank you.
Greetings, everyone. I'm Sai Subhash, currently doing complex biosystems, PhD in complex biosystems from University of Nebraska-Lincoln. I'm currently working with Dr. Jinliang Yang. Our lab focuses mainly on quantitative genetics and population genomics. Today, I'm going to talk about nitrogen use efficiency in sorghum. Nitrogen is the most essential macronutrient often supplied for enhancing the grain yields. It is the most expensive and energy-intensive input for global agriculture production. However, cereals are inherently inefficient in uptaking this nitrogen, so excess amount of nitrogen causes severe environmental issues and also economic concerns. So I'm working with sorghum. Sorghum is the fifth most cultivated cereal and also a multi-purpose commodity crop. Despite being the C4 nature, it requires high amount of inorganic nitrogen fertilizers for higher yields. So for our study, we have grown 346 accessions from sorghum association panel, which were phenotyped under high nitrogen and low nitrogen conditions. The first picture is the data that we collected from the greenhouse and also as well as from the field. We have much more data within more years and all. I have analyzed greenhouse traits here. I'm showing the 4 different traits, which is plant height, leaf count, seedling fresh weight, and seedling dry weight. X-axis is the unit of measurement here, and y-axis is the density. So we can clearly see that there is a variation for all these traits between the treatment conditions, between high nitrogen and low nitrogen conditions. So subsequently, we have used high-density genomic marker data set obtained from whole genome sequencing and conducted genome-wide association studies. We have obtained pretty much previously well-defined genes and also the novel genes by using this updated marker data set. We have updated with reference genome of version 5 in sorghum, and which is highly accurate, and we are happy to share our data with anyone who is working with sorghum, especially with the Sorghum Association panel. And my main objective of this study is to identify the genetic components underlying for different nitrogen responses. And if you want to talk more about my research or maybe about my data regarding the Sorghum Association panel, I'll be around. Actually, my poster was until yesterday or until this noon. My poster number is 48 if you want to discuss and see my poster as well. Thank you.
Everyone's been having a challenge with this, so maybe just take a look to see if it's up or down. Not that way. Hi, my name is Liz Popusky. I'm with the Kiwi Fruit Breeding Center based in New Zealand. Now, we are not implementing high-throughput phenotyping systems in the orchard yet, but that's why I'm here, because I'd love to get some really good ideas from you. So what I do want to share, though, is before we undertake this process, we wanted to understand what are the traits that we need to measure for yield components. So first, a little bit about how kiwi fruit grow and produce flowers. What are the methods that we are currently using to assess yield, and how do they compare? And finally, future directions. So kiwi fruit produce fruit on 1-year-old canes. We typically grow them on a pergola structure. So that photo is us looking up into the canopy after winter pruning. And what I want to point out is that there's variation in the size and length of the canes for each of the vines. The vines are planted about 750 millimeters apart, so they're fairly close. And also that canopy structure changes every year as you remove the cane and tie down the next one. So the 4 different methods we're going to talk about is the total number per vine. So that is just the fruit count per vine irrespective of its size. Then we'll go along with the second one and calculate fruit per linear meter by measuring the canes that only have fruit on them. The third method that we talk about is a system we call cane mapper, which gives us an allocation of where the shoots are located, whether they're floral or vegetative, and how many inflorescent groups per shoot. And the last one is the components of yield, which gives us everything that cane mapper does, but also helps us understand where we're missing yield potential with winter buds that have not produced shoots. So when we start comparing them, looking at the raw data, there's a fairly decent correlation between the total fruit number and fruit per linear meter. However, what we're interested in is a particular yield band for the growers, and that's highlighted in the red box. When we take each of the methods and go, well, how do the rankings compare between the total fruit number and fruit per linear meter? Across the whole spread, there's a really good correlation. But when we look at just what's in that red box, the correlation falls apart. Thus, total fruit number per vine is not going to be the ideal solution, and we're going to need to collect more information. This is where the next 2 systems come in, looking at cane mapper and components of yield. All 3 of these genotypes that we're looking at have the same number of fruit per linear meter, but a very different architecture type. So in the first one, even distribution of shoots and fruit number per shoot. However, as you go through, they end up with just at the apical end, which is a challenge we have in kiwi fruit. And looking at a more detailed method, this is a photo of a winter bun. You can see one of them on the left-hand side has started to initiate a shoot, while the one on the right-hand side is not going to initiate a shoot, and that's an area where we're losing potential. So what method you want to do is going to depend on the question and also the amount of resources that you have available. We would like to go forward and start to do some kind of high throughput system in the orchard, knowing that our challenge is that we are looking up into the canopy. So if you'd like to know more, please see me afterwards, and thank you for your attention.
Yeah, we're not in the slides that on. Yeah, it's really great. So it's a session. Questions?
Well, I have been challenged you a little bit with the idea that we also should push for destructive harvesting and automation of that. And I would be interested to hear if people see any chance for that, or that it's just too complicated to do that.
Mm-hmm. Yes, that was a very interesting talk. So, the automation triggered me, of course. This plant sampling robot, not the one on the slide, is a product that was developed by a company, ISO Group in the Netherlands. And they sell this robot to breeding companies to take leaf samples. But to me, we've been exploring to integrate this in our own work. But I think the main problem is, but I would like to hear your comment on it, is that if you have an enormous amount of lines developed on tomato, you can make a machine that is suitable for tomato and just take samples every day, thousands per day. But in our case, we got so many different crops and so many different genotypes and species that it's extremely complicated to develop a machine that fits such a chaotic collection of plants. And I think there it goes wrong. There is no return of investment compared to the students, PhD students that do the sampling manually.
Right. So does it apply to the fruits, or does it even apply to punch the leaves of
both? In both. Yeah. So to a robotic implementation of all the manual work that we do in the academic domain, I think.
Yeah, I can imagine that is complicated, but if you do it for one species, that would be helpful for breeders and large companies, right?
Yeah.
Yeah.
So those machines are there. So you can buy those machines. They cost maybe 100,000, 200,000 euros. I'm not a sales guy, so I don't know. But to turn it into an academic useful version,
I can imagine that's a challenge.
Yeah. So yeah, but it's more a comment actually. So
yeah, thanks.
Yeah.
Mm-hmm.
Any other comments or questions related to that? It's good.
Hello, thank you. I loved your presentation, by the way. I noticed that the analysis that you were doing was between woody plants and herbaceous plants, right? But you fitted both of them together, and from the analysis, it looks like there's a clear separation between both. So I'm wondering if you have looked at each of those groups separately and doing the allometry and allocation studies separately for each of those segments of.
Right, good question. So when you look at when you compare the young tree species, the young woody species with the herbaceous species, I found not much of a difference. And of course, this part is only dominated by woody species, so then it doesn't make any sense anymore. But if you compare the woody species and the herbaceous species at a young stage, there's not much difference in the allocation relationship. Right, there are of course many differences in other types of traits, but not in this one.
Maybe I'll just jump into this solution. Just to piggyback on the same topic while we're here, I was really interested to see the phylogenetic angle that you had, and trying to tease apart some of this variation by placing the trace onto this large phylogeny. So I noticed that there are still some outliers. There are still, and so I was wondering, have you looked at other maybe life history traits or other ecological traits that might help to further explain some of this?
Yeah, that's a very good question. So if you look at the gymnosperms, for example, you see that there are many of the species are color-coded blue. That means that they have a relatively high allocation to the leaf mass fraction. And at the same time, they have, that they have a very low specific leaf area. So those plants have a combination of a very high allocation, very high leaf mass fraction with a low LNA. And that gives a specific leaf area index if you assume a certain density of plants. And if you look in the same group to the deciduous gymnosperms, they have not a low allocation to leaves, and they have at the same time a lower leaf mass per area, or a higher specific leaf area. So that in the end ends up in a quite similar leaf area index at a given density here. So I think that it's not only purely the phylogenetic constraints, but there's also a functional constraint between the evergreens and the non-evergreens. Yeah, so there's more in that direction than.
Yeah. I guess just similarly, still in this. So trees and shrubs, they are often easy to tell apart, but sometimes a given species, you can find it with either growth form. Have you looked at species that can grow either way?
Nobody measures shrubs. Almost nobody. So there are trees in there, there are herbaceous species in here, but really the shrubs are an extreme minority. So it's difficult to say anything about that group. I expect they more or less go with the woody species, but with all the woody species, but there are not enough data to check that.
Okay.
So continuing that conversation, I'm going to ask you to speculate just a little bit. When you start thinking about allometry within the plants, you can start thinking about functional versus structural components within it. So root tips versus the rest of the roots. How would that change within the structures, and what would you expect with the allometry changes?
So you would like to separate fine roots from coarse roots?
Or expect from functional leaves?
I would expect that for function for fine roots compared to coarse roots, if you have more fine roots, you basically must have more coarse roots as well. And to function the fine roots in separate places in the soil, you also need to have your coarse roots to kind of position them in that place. So I presume that there is a kind of allometric relationship between those two, that you have to have more coarse roots if you want to make more fine roots. We had a workshop about it last week, actually, and it turns out that also there it's very difficult to find the data for plants. And if you have, there are some data, but then they are for pretty young plants, and you would actually like to know that for all the plants, right? But there's a very shortage of data in that respect. So I can't answer your question. But you would expect that there is an allometric relationship between the two.
Yeah, thank you very much, Hendrik, for your nice presentation. Before I like a lot the slide where you challenge phenotypers, saying that there is a kind of gap to bridge between between morphological traits, that is basically what we estimate with high throughput plant phenotyping, and the more functional traits that would be necessary to characterize crop functioning at the species level or the genotype level. So what do you think that we need to bridge this gap? Is only up to the phenotypers to do all the job to come through the functional traits? Are ecophysiologists who need to understand or how to benefit from the high throughput phenotyping, or there is an element probably by another scientific discipline that we have to put in the middle to bridge that gap? What is your opinion about it?
Good question. So first I want to stress, I hope that I told before that I think that a lot of these non-destructive measurements are very useful in various different settings, right? So it's only when you push from a scientific way, you try to understand these things, that I think that these high traits are really bringing us forward. Now, as far as where that has to come from, I guess that the phenotypes are generally richer than the ecophysiologists. So for them, it would probably be a little bit more easy. But I think for the ecophysiologists, they're more motivated to do these kind of things, right? So I still hope that these people can team together. And what you see that all these phenotyping systems started actually in agronomy, but they are now going into the ecology as well. So I hope when it reaches the more ecological-minded people, that the ecophysiologists there also have their push to make these things happening in the direction of the hard traits. But I realize it's a difficult thing, but I tried at least to make you aware of that there is a wealth of interesting traits that we would like to measure as well. But it's not easy. I mean, even if you measure the transpiration rate per plant, right, that is already a very nice ecophysiological parameter. But still, the plant can be small or it can be large. And we are used to kind of scale that by dividing it per unit leaf area. And I think now it is possible, if we scan the plant itself, and we know the water loss of the plants, to calculate what the transpiration rate per unit leaf area is. So I think that is what I see from here now. That is quite in reach, right? So that's something we can easily achieve. How it works when we go to these hyperspectral images and see to what extent we can determine starch and sugar, etc. That's So I think there's still a stronger gap between having the really right measurements and predict that very well. And the same is, I think, spot meters can determine the chlorophyll content very nicely. But for example, they don't look at the chlorophyll A to B ratio. And I have no idea whether that would be feasible anyway. But if you would know this A to B ratio, you could characterize what the light environment is that the plants experience. Or you have, to what light environment just. And so I think there is information where we are close, but we are not making the last step yet. And I hope that the bright minds here are able to do that in the future. Is that a little bit, it's not a fully satisfying answer, but...
Yeah, just to continue on your answer, there are some systems that predict some of your eco-physiological traits with a certain accuracy, but then nobody finds what is good enough. So if you can estimate the whole plant biomass, or you can estimate the sugar content in a leaf with a R-square of 0.6, can you use it? Or is it only good enough if you go beyond 0.8? Can you provide some guideline that you say, okay, this is useful, this is not good enough?
When you look at our paper that we wrote, R-square is a very difficult thing to use because it depends on the variation you have in the X and the Y axis, right? So the longer you calibrate with the wider the variation, the higher your R-square is. But maybe you just want to apply it at the final harvest to see what is the biggest plant. And then your R-square is not 0.99 anymore, but your R-square is much lower. Right? So maybe we should get away from simply looking at the R-square and look at what percent difference there is between the real observations and the line that you use for calibration, and focus on the percentage mistake you make or the percentage accuracy you have in that way, rather than on your R-square. And then I would say it would be cool if you have an estimate which is within 10% of the real value. I think that is generally good enough to work with. But if the difference is 20 or 30%, then it's still not so good. Is that
Okay. And Henrik, I'm wondering if to that intersection you could also add things like relative growth rate or absolute growth rates, which, because the power of a lot of the facilities that we have now is that time course series measurements that we have and that non-destructive measurement, and I'm sort of missing that relative growth rate in that intersection, because yes, we may measure pixels with one system and biomass with the other, but the relative growth rate is, often a common one.
Yeah, I think good points. I think the non-destructive systems are actually very powerful to measure relative growth rate, because that is especially a trait where if you do destructive harvest over time, you get a lot of variation, and it makes that estimate not so good as when you do that in a non-destructive system, until, of course, you get overlapping of leaves. And then if you have a rosette plant like Arabidopsis and there is so much overlap that you don't see the new leaves anymore, then your RGR is underestimated by the system you use. But before that time, I think RGR, yeah, you're right. That should be one that should be in.